Hello and welcome to the webinar on high throughput biomedical data analysis. Before I actually start up with the webinar, if you guys have any questions that you come across during the webinar, please feel free to write down it on the chat box, either of the Zoom chat box or the chat box that is present on our educational website. So feel free to put down your questions. So yeah, once again, welcome guys to the webinar. In this webinar, we would be introducing you to the biomedical data analysis techniques and the approaches that you can master and adopt to help conduct a better research and learn about the data science in the changing medical research, starting from the diagnosis to the treatment selection and development of the new therapeutic. We would also like to share with you the information and the preview of our upcoming course that will cover the most important aspects of the high throughput biomedical data analysis. That would be combining the reproducible workflows with machine learning methods and visualization approaches for various high throughput biomedical data sets. This particular course is being developed in collaboration with Pine Biotech, Sirpi.io. This webinar will be led by Dr. Anand Lakshmanan from Sirpi.io and Ilya Brodsky from Pine Biotech. Our teams are working together on new training resources for the students, researchers, and the industry professionals. In the past, we have conducted many courses and the workshops on the data science, machine learning, and biomedical research topics, including our recent sessions at NYU Langone Medical School, University of Kolkata in India, and we do conduct regular meetups in Washington, D.C., where we are helping conduct a course on machine learning with Georgetown Medical Center. You could find all the important links to the websites. It's down in the bottom, that is t-bio.info, Sirpi website, that is sirpi.io. We have learned a lot from our past experience, and we realize that it is very important to customize our training materials so as to suit the target audience. So as we share with you some of the topics we plan to offer, we are asking that you help us understand your preference and needs. So for that purpose, we have designed a survey, and the link to that particular survey was already emailed to you guys yesterday. I would be copying that link in the chat box over here again, so as to help you guys in filling up the survey. Now I would be handing over to the presentation to Dr. Anand Lakshmanan from Sirpi.io.
Hello everyone, my name is Anand. I'm the CEO of Sirpi.io. We are a company that does data science and data visualization services out of Bangalore, India. And previously I used to work at Apple in California, and I've analyzed a lot of data over the years for multiple organizations. Anyway, that's about me. And what I wanted to emphasize in the next few minutes is the power of data visualization as a tool for human intelligence, in the same way as machine learning is a tool for artificial intelligence. Now, what I mean by this is just by throwing your data onto a chart and choosing the right kind of geometry, as in a line or a histogram or a bar chart or a facet chart or a or an animation or a network chart, by choosing the right kind of chart, there is a large amount of low-hanging fruit in terms of derived, insightful human intelligence that can be achieved, assuming that we get to the point where we take your raw data, clean it up in a way that is meaningful to understand, and then take it to a level of visualization and then deriving insights. Now, this whole thing is a cycle, and it's got to be repetitive and iterative in order to do rapid prototyping and achieve your results. So with that context, let me move forward to the next slide, where we talk about data science in general. And of course, data science has impacted many, many industries. And here I'm showing two particular charts. One is the return of investment from ads, what you see on the left-hand side, where you can see that there are 4 ad sets, and there's a bunch of colors, and the more red the color, it means that the ad was a lot more effective in that time slot, where the x-axis is the hour of the day and the y-axis is the day of the week, right? So what you can see is that by a visual like this, a heat map visual like this, you can actually figure out what gets you the best bang for the buck, right? And on the right-hand side, you're seeing the market performance of multiple cryptocurrencies, and that helps you to analyze and predict the trends, and then maybe you can capitalize on your cryptocurrency investment, right? So basically, the point is that data that is visualized can help you make better decisions, because from data to visualization, visualization will bring out surprises that you probably did not expect. And it is that surprise, when put into decision and then eventually converted into action, gets you the results in a research environment or in a corporate environment, as your case might be, right? Here's an example of a visualization that's particularly interesting, where the heart rate is displayed in the y-axis, and then of course the timeline is in the x-axis. How the heart rate bump happens when a marriage proposal is made. So basically, the point is, when you visualize something, you can actually instantly find some insights just by visualizing the pattern of data and then associating the timeline with a particular task, and then find differences in the patterns, and a lot can be inferred from a chart like this. That's an example of a biomedical visualization. Let me take a few more minutes to explain why it is important to understand data science and data visualization in the context of a biomedical field, but really it's applicable to several other fields as well. When I joined Apple, the director of Apple one day called me and said that there's 4 ways to influence people: friendship, charisma, authority, and then data. And of course, data is the best way among all the other ways, because it's the most sustainable, the correct way, and it's a way that helps us relook at the data and then be open to changing our decisions should the data on the ground change, right? So that way you always make decisions from a data-driven perspective. The second important thing is reproducibility. Now, many of you are probably doing research or working in a corporate world, analyzing your own data. How do you make sure that your charts or the reports that you're generating is reproducible? What do you need to package and give it to your own community or your team? There is a new wave of research happening. It's called reproducible research, where you package your code and your raw data and your plots together when you show your research. That way, folks who are decision makers can look at just the plots, because they probably don't have time to look at other things. And then, teams that, for example, testing teams can look at the raw data and then independently verify the results that you have come to. And then maybe your own team members can look at the actual code and then verify the code or build on top of it, so on and so forth. So raw data, code, and plots packaged together in a reproducible fashion will really accelerate the pace of biomedical research. Context and comparison is very important. You probably have multiple groups in your analysis. How do you find out, for instance, the mean separation or the mean shift across groups? And what are the baselines? What is the context of the data that you're presenting? All those things can be represented or should be represented visually in order to make people understand your research, and also for yourself to convince yourself, which is really a prerequisite before convincing others, correct? Rapid prototyping. This is, and what I mean by rapid prototyping is, some of you are probably domain experts in your area, but you may not know programming as much. And some of you might be dependent on a programmer to hand over your data and then ask questions, and then they might come back to you with some results, and then you will have further questions. If you are a domain expert and a data analyst, if you are able to do visualizations yourself, you can quickly iterate from your data to a filtered data, to a transformed data, to a visualization. Maybe you derive some insights, and maybe that insights will lead you to further questions that you'll need to go back and reprogram, and then come to a better inference from your visualization. If this cycle is completed in a much more faster manner, then your progress is going to be pretty rapid. And that is why we are moving away from an old mindset where you have depth of knowledge in one area to a new mindset where you have your depth of knowledge as in your domain expertise and your data science skills. So a data analyst plus a domain expert, really makes a data scientist. And we would really like you to become the best biomedical data scientist that you can be. And then finally, communication. How are you going to present the results of your research? Are you going to write a report, or is it going to be a poster or a thesis? That's an important. Is there a way that you can take your research and then easily convert it into publishable research? Those kinds of questions come to your mind, and data science and data visualization skills have a lot to offer in that as well. So finally, when I talk about programmable visualizations, we are at a stage compared to the last several years. Several new tools have come up in the domain of data science and data visualizations, where you can programmably create visualizations, which offers you unprecedented tools in order to go back and change a particular experiment, or maybe filter and look at a different group, or summarize, or add some extra columns, or join from a different data source, or compare. If this is done programmatically, and when I say programmatically, I'm getting to a point where you can do programming without really being a programmer. In other words, tools like R programming, and there is a set of tools called the tidyverse methods of R programming that we'll discuss in much more detail during the course, and I'll also give you a flavor of it later on in this presentation. It really enables you to become a very simplistic programmer to the extent that visualizations are needed, and you close the loop in order to derive insights from your data. From that perspective, all these fantastic data science tools and context and communication and prototyping and reproducibility is really very, very important for a biomedical professional. So having said that, let me hand it over to Ilya Brodsky to take it from here, and he will talk about high throughput data in the biomedical domain.
Thanks very much for the introduction, Sahil, and it's great to see kind of the power of visualization on. And what I will be talking about is really the high throughput data in biomedical domain, because up until now we see the power, but when we talk about the type of data that we have, it becomes untrivial as to how to really visualize in a powerful way the type of data that is emerging and exponentially increasing in the biomedical domain. So let me give it a little bit of an overview, a history of genomic data, kind of how did we get here? So in 1950s, the DNA double helix structure was discovered, and it took about 40 years for that data to really impact the way that we view health and disease. We started to understand that, it has a tremendous impact across all areas of biomedical and, in fact, biological research. The two people that spearheaded that project, Francis Collins today is the director of NIH. Craig Venter is the founder of Human Longevity that uses integration of multi-omics data to address the challenges of aging. And so you see that those ideas have a tremendous impact since then on what we understand as the drivers of human disease. But it took another maybe 3 to 5 years for that data to become massively available and affordable when Illumina started their sequencing business. And today, since then, so it only took another 9 to 10 years for us to have a lot of data available. So in 2015, we had the final phase of the 1,000 Genome Project with over 1,000 genomes being available, fully sequenced human genomes. And today, in 2018, I think we can all appreciate the amount of high throughput data that is becoming available. So the MIT Broad Institute already sequenced over 100,000 whole genomes, Genomics England over 50,000. And of course, this is whole genome. So you can imagine that a lot more is available for other types of data. And as a result, we now understand that the disease symptoms that we used to rely on clinical data to measure today are really defined by the underlying layers of what's called omics. So we have the genomics, the proteomics, transcriptomics, epigenetics, microbiome, a lot of data, and each data set here is huge. So let's take a look at an example data set. An example data set that we'll be using during our course and during this short presentation is taken from a publication in 2013 where a number of different cell lines were taken from breast cancers and sequenced for their epigenetic changes or variation, for their genomic changes or DNA, and for their transcriptomics, for their gene expression. And all of that data was used to predict drug response for different subtypes of breast cancer. Now, to deal with that kind of amazing quantity and high throughput qualities of data, we have prepared a platform that provides easy to use access to multi-omics data processing and integration. And during this short session, what I will be showing you is how can we go from the raw data, which is short reads, to a table of expression, which now starts to resemble the type of data that you can actually visualize. So the section that we will be looking at is transcriptomics, right here, RNA-seq. Probably a lot of you are familiar with RNA-seq. And really what it does is it takes short reads and transforms them into something that we can start comparing between different conditions. So how do we process this kind of data? Well, you used to have to know Linux, and you had to, upload these huge data sets onto your computer, and your computer had to be very powerful, and you had to monitor the process of data so that you transfer it from the right folder to the right folder and automatically build pipelines. And we decided to address that challenge of the complexity of the processing and prepare this intuitive interface that simplifies the whole process to a point-and-click interaction. And as you select each new button, each new step, you actually can learn about it as you walk through the pipeline creation. So for example, the first section, you would use some pre-processing to reduce the amount of replicates that don't represent the biology, that are really technical. You then can use very complex statistical analysis of reads of how they fall onto the genome using bowtie, for example. And then finally, prepare a comparison between two conditions or more conditions. But as a result of this pipeline, really what you get is a table of expression, just like this, where you have gene names as your rows, and sample names as your columns. So the samples are going to be names of different cell lines. And of course, these are breast cancer cell lines. So you would have different names of subtypes of breast cancers. And now you can identify the differences between numbers to understand how these numbers can give us insight about the differences between individual cell lines and groups of cell lines that could be compared. Now, how do you deal with this data? And what's the problem with dealing with this kind of data? Well, first of all, it's huge. You have between 20,000 and 60,000 genes for each sample. So imagine between 20,000 to 60,000 rows in your table for however many samples you have. And how many samples do you have? Well, you can have tens to hundreds of samples because we have so much data available. If you go on databases like NCBI, ArrayExpress, and others, you can really see that that today data sets contain multiple, tens to hundreds of samples. And to deal with that, you need high performance computing. And that's why our platform is on the cloud, and you don't have to upload or download any samples. You can do all of your analysis there to prepare it for something that you can do on your computer. Now, the other challenge is that these are very complex relationships, right? So what we saw when Anand was presenting his data, ROI for ads, or even tracking the performance of markets, or using something like a heart rate to visualize that data, it's intuitive. We know what heart rate is correlated to. We know when our heart beats higher or lower. But with gene expression, I mean, there are so many pathways that interact in complex ways that have different signaling or actual functional differences between them. To know all of that, it's impossible to hold it in your head, so it's not intuitive, and you really have to have additional tools to deal with that kind of complexity. And of course, it's non-trivial to interpret that kind of complexity, right? So therefore, you really have to rely on data exploration and visualization in a programmatic way. And you have to utilize supervised and unsupervised machine learning, which is a component of what we will show you today and what we do in the course in a much more expanded way. So how do you go from a table that contains all of this complexity to a simple visual scatter plot that can show you the differences between, for example, here, luminal and other types of breast cancer? How do we do that? Well, just in the same way. We can use the platform and upload and have this huge data set already on the system so that we don't have to rely on our personal computers. And then we just use this simple chart to create a principal component analysis. And this is an example here on the right that you can see rotating a number of dots in a multi-dimensional space, reduced here to 3 dimensions, but in reality it's multi-dimensional. And you can see how turning this scatter of dots can actually help you see that some angles show differences between different groups much better than others. And that's what PCA does. PCA actually will take the table and rotate it in such a way that we can separate it into a 2-dimensional space, so to flatten it, and make it much more simple. So before we go on, I am just going to quickly demonstrate how to do that. So what I have here is the platform, the t-bio.info platform. And you can see that we have the raw data processing right here. And what I will do is go under unsupervised analysis. And you can see this pipeline that I just showed. And I'm just going to upload a file. So I have the cell lines expression data right here. In fact, I'm going to use the marked where I have the marking of the different subgroups. Continue. I press start. And as you can see, every time I click, I can learn about what this procedure does. So here you can see that the principal component analysis is a statistical procedure that uses an orthogonal transformation to convert a set of observations of possibly correlated variables into a set of values of linearly uncorrelated variables called principal components. Okay, so I change the dimensionality and I can then visualize it in a much more simple way. So I click on end, save this, PCA, gene expression example. And during the course, by the way, each one of you will have access to the platform for the duration of the course. And now I've run this analysis. Now I'm not going to wait for it to finish because I already have a prepared pipeline from before, download pipeline output, and what I get is a PDF. So I'm just going to download this PDF, and I can take a look at the sample separated between luminal, normal-like, and then basal and cloud and low. Now I have some insight into the data, some basic idea of what the data contains, and what I will do is I will actually hand it over back to Anand to talk about how do you improve this visualization to make it much more informative for downstream analysis.
So now let's go to hands-on on data visualization, right? So for this hands-on data visualization part, I'm going to go to a platform called the RStudio IDE, where the screen that you are seeing here is on the left-hand side is a set of programming. We'll actually go into much more detail in the programming in the actual course. But what I want to point out here is how do you go from the output of the pipeline and run this principal component analysis and take the results, and you see this percentage greater than percentage. That actually means the pipe, which basically means you take whatever is there on the left-hand side and pass it on to the right-hand side, and think of it like a stage-by-stage cascade, and then get to a plot, and you see a plot that's something like this. What you see in this screen is how, so let me first explain the different colors. The different colors represent the different groups, so basal, luminal, normal light, so on and so forth. And each of these data points comes from the sample, and you see these lines. Those at the end of the lines, the actual gene is indicated. The label is given here. So, for example, let's say we take this particular gene, ENSG 000000160-180. We can see that in this particular direction, this gene is responsible for the maximum separation of this group and this group, right? This group is the luminal versus the other group is normal, like, for example. Right? So what you see in front is a visual way of representing the cluster of data points colored in specific ways that enable you to see the differences between groups. And not only that, to be able to identify that particular gene which is responsible for the most separation between the groups. And therefore, we can actually identify which gene is an important player in this. On top of it, you can actually, this is an interactive visualization, right? So so let me clear the annotation and then show you that you can, for example, as you hover over each data points, you can actually see the variable values and the groups. And if you're particularly interested in let's say one of these, you can actually zoom in into a lot more detail. Because when you zoom in, you're going to get like individual level data, and you can programmably make sure that you can display whatever you want in the tooltip. And therefore, it helps speed up your, it helps speed up your own research process. But also it's important to zoom out and then see the overall picture, because overall picture gives you a much better insight in ways that zooming in does not give you. As a general principle of data visualization, this ability to zoom in and zoom out is very critical when it comes to research, And I would recommend that you keep Google Maps or Apple Maps in mind, wherein, for example, if you're using the maps, it is a pretty complicated map with a ton of information, roads and satellite data and traffic and buildings and whatnot. But yet, it gives you information about where you are and where you want to go and the 2 or 3 paths in front of you, right? So you can zoom out and then figure out the overall strategy, and then you can zoom in and then take the next step, as in, turn right, so on. Right? So in other words, if we are able to have that analogy in mind and then come up with an overall picture by zooming out and then also zooming in to do individual analysis of each data point, right? So that's what this this visual represents. So now let me go back to the presentation. So I guess that's what I wanted to say, and you can go and I think I'll pass it on to Ilya to explain this chart in the context of raw data. What you're seeing here on the left-hand side is the actual principal component analysis. And now that we know that this particular gene is important, we can go back to the raw data, which is what's shown on the right-hand side, and Ilya can explain what we should look for in the raw data, given the new insight that we got from the principal component analysis. Over to you, Ilya.
Thank you, Anand. So yes, as you can see, visualization helps me identify some trend in the data. And like we said before, it's untrivial that this gene, for example, has some role in separation between luminal and the other types. Now, the best difference that this principal component shows us is indeed between luminal and basal, or luminal and normal-like. But we know from biology that basal and normal-like are fairly similar. So you can see this on the PCA, they're closer together. And in fact, on the right side, if you take a look at the level of expression, you can see that the majority of the luminal are upregulated or highly expressed, and the ones on the right side are all low expressed. And so you can see how this difference, if you imagine having a table of thousands of rows, how do you find this one gene? Well, that's where machine learning really comes in to help you extract these things and then help you visualize and explore your data so that you can interact with it. So now you can see that this is the gene, and these are the 2 groups that we see the biggest difference, and these are actually the different subtypes that we can interpret the biological meaning of this gene expression. Now, all of this was a little preview and an explanation of what the types of things that we will do during our course. So the course that we are about to launch as a joint course between Serpi.io and Pine Biotech is to take this high throughput data, genomics, transcriptomics, epigenomics data, and address the challenges of high throughput biomedical data, how to process, how to use machine learning, how to use visualization, and really give you hands-on experience that will combine some user-friendly tools like I showed you on the platform, and some programming, simple programming that you can incorporate into your workflow so that your practical skills in R and using user-friendly tools give you ability to visualize and gain director insights, and hopefully make your research more reproducible, also navigate the data so that your research is data-driven, and bottom line, more impactful. When you incorporate these kinds of skills into your workflow, really the result is that you will understand the methods, you will understand the high throughput challenges in biomedical data processing, you will understand how to approach analysis and how to visualize your results. And inside the course, actually throughout the course, we will use several different projects that are relevant to clinical applications. I mean, how do you use genomic data in the clinic? In research, how do you understand the basic biology behind these genes and behind these samples that we have? And even pharmaceutical topics. How do you use gene expression data to design new therapeutics, to identify new targets? How do you use it to reposition drugs and repurpose different drugs? You will be able to prepare and plan your own analysis. As Anand was saying, you will be able to think and convince with data and not just with words and imagination. And finally, you'll be able to really incorporate all of this into your own portfolio and your resume. So you will be able to stand out from the crowd when you go and apply for your position, or when you want to advance your own career in your current position. You can claim having more skills and more capabilities. And I want to touch on one other point that maybe is hasn't been highlighted. It's important to understand that today's publications are changing from printed compilations of results into something that somebody else can take and follow your steps and validate that indeed what you're saying is true, but also explore other things. And, I'm talking about others, but I want to talk about your research, your publications, your work. You want to be able to improve your work by comparing it to what other people did, and therefore you need to understand their methods. By understanding what their data says, therefore you have to be able to rapidly go through other people's data and intuitively understand what it means and where are their correct assumptions and incorrect assumptions. And then you want to actually publish more impactful things, even without the need to generate a lot of your own data, because of course so much data is available today. So these are some of the topics that we will be covering. Anand, I don't know if you want to add anything to these points that I just made.
Yes, Ilya. So I want to add a couple more things. One of the very important takeaways from this course that you will get is a transferable skill that can be applied throughout your career, whether or not you stick to the biomedical field later on. People switch careers all the time. And if you, get data analysis and visualization skills, pretty much there is no field today that does not analyze data. And fields like finance, for example, have taken this algorithmic processing of financial data to a completely different level, and they are like banking on it in order to do investments and analyze portfolios, so on and so forth, right? Several other tools are being invented across domains, but the ability to visualize step by step and use visualization as a diagnostic tool for your data quality problems, for your data wrangling and transformation issues, your machine learning, visualization of machine learning, explanation of your particular procedure, that transcends the biomedical field. So while you can do a better research in terms of your own biomedical data that you're following, what you're learning is something that will follow you throughout your life is one point that I wanted to infer. The second thing is both SIRPI and PINE, we are dedicated to education, and we will take we will go the extra mile in terms of connecting with you to make sure that you understand the problem that you have in front of you from the context of either domain expertise, in which case Ilya will help out, or if it comes to visualization of your pipeline results and learning simple R programming for visualizations, in which case SIRPI will come and help you. So that's what I wanted to add to this, and over to you, Ilya.
Okay, yeah, I think those are great points. And at this point, we do have a little bit of time for questions. If you have any questions related to the examples that we showed or related to the upcoming course, please go ahead. You can either write them in chat or you can unmute yourself and speak up.
I think there are some questions also that perhaps Sahil can if Sahil, if you find any questions that's written, if you can speak out those questions. One of us can take those questions if there is
Okay.
any.
Sure. Yeah.
Okay, I see here, will the course focus on what various ML algorithms we can develop or just follow the existing pipelines available? Sruti, thank you for your question. I think Sahil already said that, we will touch on both aspects. In reality, what you see today, and this is actually a very interesting topic, that machine learning started emerging as a fairly rapidly increasing topic in biomedical research, starting about the same time that genomic data started becoming more and more available. And it's obvious why. You have explosion of data and change of data. You can't rely on simple and intuitive interpretation. So you refer to other tools that do it automatically. And surprisingly, in 2015, that trend started changing for several reasons. One reason is that diversification of machine learning tools. You start having deep learning, you start having neural networks, you start having classification, new methods that started appearing in classification. Unsupervised machine learning is actually called data mining a lot of times. So you really have this kind of, if you think of a tree branching out of machine learning, which a lot of times people refer to, things like PCA, like we showed today, as your standard machine learning tools, into much, much more variety. And today, when we talk about developing your own machine learning methods, really what we mean is how to effectively combine these different methods and modify them so that we can do feature engineering, so that we can do pipelines of machine learning methods, so that you can arrive at more impactful results. And that will be the focus of our course. We will not actually touch on algorithmic development, but we will touch on how to optimize and how to appropriately select the right methods for the analysis goal. We will touch on feature selection and feature engineering to a level that you can apply it to biomedical data. And of course, we will do a lot more on the data visualization side, so that the results of machine learning that oftentimes, just kind of pop up and, become like a black box. You don't know why you arrived at this conclusion, so that it's more clear. You can actually go back and understand what is the reason why these things were selected.
Let me add.
Yeah, go ahead.
Sorry, let me add one more thing to this. So for the input of your machine learning models, the quality of data is critical. And for this particular, in this platform, if you're using existing data and if it's already cleaned up, that's one thing. But if you have other data sets, and if you want to get a handle on how to clean up the data and how to use data visualization as a diagnostic tool to understand data quality problems, for example, using genome density, and then finding out whether your data has, extreme outliers, and if so, do they make sense, understanding the bounds of the raw data to evaluate the quality of the raw data as the input feed for your machine learning models become crucial in order to have increased confidence of the output of your machine learning models, if that makes sense. So we'll talk about data visualization in the class to be able to clean up the data and then have a much more higher quality input stream of data for your ML models.
Thank you, Anand. And here's another question from Deborah, and I want to address that as well. So she's saying, I have no prior experience of next generation sequencing, though I have worked on capillary sequencing, and hence how this course is going to help me, as I am a hardcore molecular biologist working in R&D of a company. So, I'm going to address it from my perspective, which is more on the data side and less on the wet lab side. But I do want to say that today, biology is obviously very data driven. I mean, if you literally pick up any publication, you will have a method section, and the majority of them will use some level of statistical analysis, obviously, and a lot of them will incorporate machine learning. A lot of them will refer to data. A lot of them will give access to the data. And I think that, a lot of that is driving more effective R&D. And I can give you a practical example. So practical example, we are working with a lab that is sequencing a lot of data from infectious diseases as they are being, as a certain therapeutic is being tested on mice. And you have a lot of variables. Obviously, the mice get sick and then, they're sacrificed, they die. And you have to understand, well, how do I measure the effectiveness of this new therapeutic that I'm developing on this specific disease? And you have a lot of variables to consider, right? The individuality of the mouse, the different proportions, size, etc., health of the mouse, the different time points when you infect the mouse, the different amounts of this compound that you're testing, the place where you inject, and these are just on the surface, right? So these are on the surface, but deeper down, you have so much variation that is on the omics level that the question becomes, well, how do you make this process more efficient than just trying something because you think it should work intuitively? And the answer to that is that you have to refer to the data to drive your next experiments. You have to have access to the data. You have to be able to integrate that data on some level. And you have to really identify where is signal and where is noise. And how do you use that signal to plan your next steps? And that's a little bit of what we will try and focus in this course. We will take several examples that incorporate this kind of data to arrive at some conclusions that are relevant to R&D in biotech companies, that are relevant to clinical diagnostics, that are relevant to understanding of the basic biology on some level. We're not going to do a molecular biology course on, full-fledged interpretation of results, but we will utilize some tools, like annotation, pathway annotation, etc., so that you can at least have, an ability to move in that direction. And maybe this is relevant to what Manny is asking, what data sets or types of biomedical problems we will be addressing in the course. So I referenced one example. The one example that we used was essentially a collection of cell lines, different breast cancer cell lines that we have multi-omics data available for. And those include genomic data and epigenetic data like ChIP-seq, and gene expression data, which includes gene expression, isoform expression, RNA editing, etc. Yes, I would say that, a lot of this is going to be focused on high throughput biomedical data, which will be omics. And that's not just, transcriptomics, but it will incorporate, NGS. And how it's relevant to phenotypes, right? So the phenotypes are going to be, in this case, in the case of the project that we looked at, is actually data from screening, so IG50, inhibition of cell proliferation, and how do you find the correlation between those data and the signal from omics? And how can you combine the two to predict the effectiveness of specific compounds that were tested on those cell lines? So that's going to be one example. The second example that we will use is a data set from a toxicology data set. So basically, there are many compounds. Some of them are known to be toxic. Some of them are known to be non-toxic, right? Therefore, more useful for designing different therapeutic compounds. And so the question is, how can we differentiate between the two? How can we identify early on some indication of toxicity based on descriptors of these molecules? So no next generation sequencing data, but the data is going to be biochemical. So it's going to be using the physicochemical properties of these molecules, and we will learn how to use machine learning tools to differentiate between the two and to understand what is the relationship between the physicochemical properties and the final effect of that molecule. Okay, another question here from Benedict. Which machine learning algorithms that applied most in biomedical data? Again, I think it's I can actually share with you, if you want to shoot me an email, a very nice publication that did analysis of recent biomedical publications and the utilization of different types of algorithms. So we have, you know, 3 or 4 different categories of machine learning, right? So, for example, you can separate them into supervised and unsupervised, right? So unsupervised would be things like clustering and dimensionality reduction, and supervised will be classification, network analysis, etc. So what is the difference between the two? Well, the difference is primarily you would use one to mine your data. You're looking for trends in the data. And the other one will be more on the, okay, how do I use a trained data set to predict some quality of a new data set that I've generated? And so in biomedical field, you can also start thinking about the types of problems in biomedical research could be separated into 2 categories like that. Sometimes you want to, you know, explore the data. And sometimes you actually want to use some results of an analysis to help you, you know, for example, apply those classifications to a new data set. So to answer your question, though, more directly, PCA has been used since, you know, the beginning of genomic sequencing extensively and in a growing way up until 2015. More recently, people started using TSNE or TSNE for dimensionality reduction, for example. And primarily that's because of single cell sequencing, where it's become much more popular. So, you know, those are 2 examples of very useful machine learning methods that have been widely popular and used. And not only that, also used to support different classifications and different clustering techniques, etc. So I don't think there's a list of very, you know, most, what did you call it? They applied most. But we will touch on the different categories and the different types, and of course see what is their use, so that you can appropriately either use them as is or modify them in a pipeline, or modify the data that you feed into those algorithms, like Anand said, by filtering, by feature engineering, by preparing the data and making sure that the data quality, because bottom line is garbage in, garbage out, right? If you don't feed the algorithm with good data, it doesn't help you if the algorithm is good or not. Okay. I don't know, Anand, do you want to add anything to these questions? Okay, well, so again, it would be great that if you have any further questions, you can actually email us directly. You can also write those questions in the survey. To register for the course, so let's just go on to the next slide. In the course itself, we will publish the full curriculum of the course based on your survey replies. So if you want us to consider what you think is the most interesting topic, I mean, we are not limited to the things that we discussed here. These are just some examples, and we're still in the planning process of the course itself. and of course, you are welcome to help guide us so that we can more appropriately address your needs. Okay, final question. Should we be trained on how to arrange reads generated from NGS followed by its analysis and interpretation? So Deborah, we will actually practice that. We'll not go into the algorithmic or statistical detail, but what I showed in the beginning is that can be a very intuitive and simple process, and we will actually cover a lot of that in our course. So, you know, if you don't know a lot about processing of next generation sequencing data, we will do that for you. I mean, we will train you how to do it, and you'll be able to practice on several data sets, so you get a better idea of what combinations of methods work best, and what are the outputs, what are the inputs, etc. Again, we have other courses available on our platform on edu.t-bio.info. That you can actually take full courses on NGS, on transcriptomics, on genomics, on metagenomics, and you can, you know, those are free of charge. You can take those and try those out if you want. I do want to mention, you know, there were some questions about machine learning. We do have a course that's available. It is a paid course because it is being done for Georgetown Medical Center. Jointly with them. But if you're interested in machine learning for next generation sequencing for high throughput data, go ahead and take a look on edu.t-bio.info. We do have a full course on machine learning. It is right now being recorded and put out, so you can definitely enroll because the first lessons are already available, but the full course will be completed by the end of October. And so this is a perfect time to join. So just as a final note, we do have more resources available on our site, t-bio.info, and serpi.io also does training workshops on visualization and scripting in R, and you should definitely check out their website, serpi.io. We will be able to either enroll you in this course that we are jointly developing, but you will definitely benefit from looking out at the existing resources and joining one of the separate workshops that we are conducting. Anand, do you want to add anything to this?
So I just want to add one quick thing that from sirpi.io perspective, the vision of the company is to have data science and data visualization at the core, and then identify specific domain areas and collaborate with folks in different areas and come up with joint courses. So to that extent, you know, we have the course called Data Science Using R that we developed internally as an independent program. You can definitely check it out at sirpi.io/courses, where it will be a little bit more programming intensive, but still geared towards domain experts without having the need to go through like complicated programming constructs, and still be able to import, wrangle, visualize, transform, summarize, and visualize and model and get results out of your data. So that's a full course by itself. And then we have other courses like data science for equity market analysis, if you're a finance person. And then, of course, with this joint course with Pine Bio that we are really excited to give out. And if you have any specific needs, we can also customize certain courses to you know, groups of you or individuals or for corporates or for universities. Especially this new tidyverse methods of pipeline programming in R is something that we are really passionate about at Sirpi, and we'd be more than happy to engage with your individual needs.
Yeah, I think it was great to you know, do this together, and hopefully we will send out an email update about the other plans that we have. So please stay in touch by filling out the survey. If you have any issues with the survey, you will also have our contact information, and of course we already have your emails. So look out for that update from our side as well. And again, happy to you know, get to know some of you and answer some of these questions. So thank you very much.